Wang presenting on concussions, but Mr. D, are there any announcements?
I've got none.
Okay, so here's Dana presenting on concussions.
Oh, I should pull it up.
First time presenter.
No, that's not how that works.
Yeah, it is.
Dana's a first time presenter.
So what?
So you can ask questions about clarifications, but no, like, yeah.
Oh my God.
Should I not drink this? I won't drink it. No. I'm not good.
Okay, so hi guys. The title of my paper is Candy started the angiotensin to AT1 receptor blocker and PPA or gamma angioist reduces lesion volume and improves motor and memory function after traumatic brain injury in mice. So that's a really long, complicated title, but in English it basically means blood pressure drugs helps mice recover after a concussion. So obviously so many people helped me with this presentation. It would not be possible without the help of so many. But I want to say a special thank you to my fellow Project 80 heads, Gabby Stern, Max Leaf, and Kim Chen, because they have helped me in so many ways, so thanks guys. Okay, so on Monday you guys all saw Project 80's latest video about concussions. You guys might be wondering, why are we spending so much time on this topic? So unfortunately, I'm sure all of you know at least one person who has gotten a concussion. I know a lot of people, I'm sure there's multiple people in other grades who have also gotten concussions. So it's turning into this really big problem in our community. But even though we have so many people with concussions, we don't really know what they are, how they affect our lives besides, oh, you're not really supposed to play sports after them. But we don't know how they impact other parts of their lives, like academics. So this field obviously needs a lot more research. So it's already known that increased blood pressure after concussion is really bad for concussions, because with increased blood pressure it leads to more swelling, and more swelling in a concussion is bad. So this particular drug works under two molecular pathways. It blocks the angiotensin II pathway and it activates the PPA gamma pathway. So in order to work, it has to block this pathway and it has to activate this pathway. Does anyone have any questions about that so far? No?
Wait, can you say what those pathways do?
Oh, okay. So when angiotensin II is overstimulated, it leads to more swelling. So it makes sense that you want to shut that pathway down. And PPA gamma, it has to do with like fatty acid storage and glucose metabolism, but the activation of this pathway is what leads to the drug's effectiveness. Everyone good? So the experiment. The mice were basically split into three groups. There was a control group, there was a sham surgery group, and there was a controlled cortical impact group. And so in the sham surgery, the mice were put under and their skulls were open, but their brains weren't actually touched. And in the controlled cortical impact group, that was when they got surgery to mimic a concussion. So basically the brains were injured in a specific way to mirror a concussion. And then in each of the three groups, the mice were split in half, while half of them got the vehicle or the placebo, which is basically nothing, and the other half got the drug.
Dan,
Yeah?
how did they do the control cortical impact?
There was they had like specific measurements of like where they injured the brain and stuff. I can show it you later.
They like hit a mouse in the head with a hammer?
Well, I think they took like a scalpel and like kind of, yeah. So, okay, so overall the drug had really positive, really beneficial effects on all the multitude of concussion side effects. And what was really interesting is that the drug actually helped on both the molecular level and the physical level. So researchers saw results both at the molecular level and physically they were able to observe results. So I'll be going into more detail about all of these. So the first one, decrease in lesion volume. So after concussion, your brain hits your skull and the injured parts of your brain are called lesions, and they're here in these red dotted areas. So this data is from 3 days after the surgery, and this data is from 28 days after the surgery. So this mouse just got the placebo, so nothing. Drug, placebo, drug. And as you can see, the red dotted line on this one and this one is a lot smaller than when compared to the control, which means that the drug helps reduce the injured parts of the brain. So it helps make these lesions a lot smaller. And the same information is shown just in numerical, graphical form down here. And then decrease in neuron death. So the neurons, the cells in your brain, they start dying once the brain is injured. So what the researchers did is they stained all the dying cells green. So the green dots are the dying neurons, and the red dots are just the leftover neurons. So this mouse got the placebo, drug. So can anyone tell me the difference in color between these two pictures? Yeah, there's a lot less green in the mouse that got the drug, which means that the drug helps prevent neuron death. So the same thing is shown in these two graphs, and the y-axis is the number of dying neurons. So if it's a higher bar, that means more neurons are dying, more cells are dying, which is bad. So the drug is preventing these cells from dying, which is a good thing. Okay, so after concussion, one of the physical symptoms is that you start having issues with your motor function. So the researchers wanted to see, could the drug actually help with the physical side effects as well as the molecular ones? So they did a rotarod test, and that's a picture of a rotarod. It spins, they time how long it takes the mice to fall off of this. And so they trained the mice on this 1 day and 2 days before the surgery, and then they tested them 1 day and 3 days after the surgery. And they found that the mice that got the drug stayed on this spinning rotarod for a longer period of time than the mice that just got the placebo. So this drug actually helped the mice with their physical side effects and not just the molecular ones. So the researchers can actually see these results.
What are all those groups?
There's the control vehicle, control drug, the sham surgery vehicle, sham surgery drug, the controlled cortical impact vehicle, controlled cortical impact drug. So that's why there's 6 bars. But, yeah. Okay, and another physical side effect after concussion is issues with your memory. So the researchers decided to run a Morse water maze, and that's a picture of one. So basically you fill a tank with water, and then you put this little plastic platform in one of the quadrants. And the researchers started this on day 24 after the injury, so they let the mice swim to the plastic platform, and then on day 28 they took out the plastic platform, and then they timed how long the mice spent in each quadrant to see if they basically remembered where this platform was. And they found that the mice that got the drug they spent more time in the correct quadrant, which means they kind of remembered more where this plastic platform was, which means the drug was helping their memory too. We got that. Sophia.
How many mice were used?
The researchers didn't specify.
Wait, so in the first 2 columns that said that
This one?
yeah, the vehicle had more memory, or?
Well, yeah, but I guess the researchers didn't really think that those results were big enough to cause concern.
Oh.
And this was the sham surgery group, so they didn't actually get the surgery that mimics a concussion. They just had the skull.
Oh.
This is the group that actually got the surgery. So.
Was your question being answered?
Yes.
Yeah.
Okay, sorry. Okay, so as I mentioned before, this drug actually works under 2 molecular pathways. And this is the second pathway, the PPAR gamma pathway. And this pathway gets activated by the drug. So the researchers decided to test what would happen if they deactivate this pathway, what would happen if they tried to turn off this pathway. And they did this by using a PPAR gamma antagonist, which basically turns off this pathway. So then they had 4 groups. They had a control, they had just getting the drug, then they had just the antagonist, and then they gave the drug and the antagonist. So basically they're trying to see what would happen if you turned off this pathway but still gave the drug. And they found out that once you turned off this pathway, basically the beneficial effects of this drug were really diminished, they were really lowered. So this pathway getting turned on is really essential to this drug working. And over here they did another rotarod test to see if the physical effects were also changed, and they were. I know this is a really complicated slide. Does anyone have questions on this one? Okay, good. Okay, so what's really out there? So obviously, candisantan has been established as a blood pressure medication for humans, but hasn't really been established as a concussion medication. So obviously more research needs to be done for that. But also just speaking to concussion research as a whole, right now the only real protocol we have is go home, get some rest, try not to watch too much TV, but there's really no treatment. It's becoming such a big problem, especially at Pingree when we have so many student athletes. There really needs to be some sort of safe, effective treatment that athletes and students can follow to hopefully safely recover. So also watch out for Project 80 put to concussion commercial. So thanks guys. Does anyone have any questions? I can go back.
Can you go back to the last graph?
This one?
Yeah. What's being shown on the right there?
On the right? They did another rotor rod test. So they did another spin test and it's latency to fall to see how long it took the mice to fall off.
Okay, and this is the one where they're basically...
This is the one with the PPAO gamma antagonist. So they gave just the drug, they gave the antagonist, and they gave the drug and the antagonist.
Okay.
This one, this is, they were testing the amount of immune response cells. So this is the y-axis. So if there's more immune response cells, that means there's more swelling happening. So a higher bar means more swelling. Yeah.
Did the mice have any side effects from the drug?
If they did, it wasn't mentioned by the researchers. Yeah.
What do the third and four bars on the first graph represent?
These three?
Yeah.
So this is, they gave the controlled cortical impact. So all of these got the controlled cortical impact. So they all got the surgery to mimic a concussion. And this one, they gave nothing, vehicle, placebo. This one, they gave the drug. And this one, they gave that PPAO gamma antagonist. So they shut down the pathway in this one, and they activated the pathway in this one.
Can you go back to the lesion slide?
Wait, did you have a question on this slide?
On the right slide, what do we call each other?
So there's 5 groups in this one. They gave one sham surgery with control, so nothing. So they just opened the skull, gave nothing. And then the second group is they gave the surgery, but they gave them a placebo, so surgery but nothing. Then they gave surgery and the drug, surgery and the antagonist, and then surgery, the drug, and the antagonist. Okay. You got that? Yeah, higher bar. Oh, sorry. A higher bar means they took a longer time to fall off. It was the spinning rotor rod, so they took a longer time to fall off. Coach Scott.
Was there any mention to healing time of the lesions, or was it all symptom-based? Like, did the brain heal quicker than the mice on the medicines, or were they just not showing the symptoms associated with the injury?
Well, I guess you could say they did heal quicker because all of these things were improved by the drug, but yeah, so I don't know that. So you want to go back to lesions?
Well, I think our question is kind of the same.
Yeah.
If you kind of go over just the basic graphs, the lesion graphs specifically one more time, I think it will help.
Okay, so this is from a mouse 3 days after injury who didn't get the drug at all. And this is from a mouse 3 days after injury who got the drug. And the red, I don't know if you can see from back there, but there's a red dotted line outlining the lesion, the injured part of the brain. And there's one up here too. But this one is a lot smaller than this one, which means that the injured parts of the brain were healing a lot quicker with the drug. And this is only 3 days after injury, and this is 28 days afterwards.
And this is which lesion? This is the surgical?
Yeah, this is when they actually did a surgery to mimic a concussion. Because the other mice didn't have lesions because they didn't get injured. Yeah.
You know why it seems to get worse after time goes on? It looks like the dotted line is bigger.
I have no idea.
Where are you?
You know, like if you compare the two, like the 3 days post injury and the 28 days, it seems to get worse over time.
The graph includes the lesion percent volume is smaller. Just the picture.
Yeah. The graph is the same information just in numerical form. So this side is the mice who got nothing, they got placebo, and this side is the mice who got the drug. And this is lesion volume, so it's measuring how big the lesions are. And so this side is higher than this side, so they had bigger lesions without the drug.
I had no idea mouse brain could regenerate.
You learn one new thing a day.
And then the cell death, can you hear that one?
Oh, so good, sorry. So the green dots are the dying neurons, and the red dots are just regular neurons. Yeah.
How exactly they died?
They said they used this tunnel plus staining. Stains them green. Yeah.
So, they took some photograph of some sort, correct? I mean,
Yeah.
we're looking at some representation. How much of the total area are we looking at?
That is a very good question.
I mean, are they looking at just a very small area of one and a very small area of another?
Yes.
I could look it up and get back to you.
Okay.
So, yeah.
When are the two, like the PPAR gamma and the other pathway, like activated under normal?
So angiotensin 2, when it's overactivated, it leads to. Oh, you're saying when it gets activated?
Yeah, like are they not like in?
Well, I know PPA gamma, it gets activated when it's regulating fatty acid storage and glucose consumption, and I just know the overactivation of the other one leads to more swelling. So, yeah.
Do you know why the cortex and the hippocampus, because like different parts of the brain, were tested in this decrease in neuronal death?
I guess the researchers want to see what would happen in different areas of the brain, not just one, to see if it was like overall.
Hippocampus is memory.
Yeah.
And cortex is the motor skills. So the two symptoms you're talking about memory loss and motor function. That's why that's.
So really this is focusing on the blood pressure and...
Yeah.
Is there any reason why like one of the bonds is far higher than the others?
This one is for higher. Well, this represents different areas of the hippocampus, so. Yeah. Yeah. Thanks, Andrew. Is there anyone else? I can keep going through other slides now. Okay. Well, thank you guys.